Welcome! Today, we're going to tackle a really fundamental question. How does our brain actually build our sense of time? And how does it weave that sense of time into the fabric of our memories?
Okay, so here's what we're going to cover. First, we'll dig into how the brain even perceives time in the first place. Then we'll introduce a really elegant theory of memory called the factorization hypothesis. After that, we'll look at a couple of big challenges it faces, and finally, we'll explore two potential solutions that neuroscientists are working on.
So, first things first, we have to get our heads around a basic tension. And that's the difference between time as a clock measures it and time as we actually feel it passing.
You see, here's the thing. Our brain doesn't have any time receptors. There's no organ for sensing minutes and seconds. Instead, it has to infer the passage of time from change. And that leads to a totally subjective experience, one that can feel like it's speeding up or slowing down. And this subjective experience follows a well-known principle called the Weber-Fechner law. What this means essentially is that our perception scales logarithmically. A 1-minute wait feels a lot longer when you're 5 years old than when you're 50. It's all about proportion. So that brings us to the next question. How does the brain store memories that are so fundamentally tied to time? How do you remember what happened, where it happened, and when it happened?
Well, the brain makes a really important distinction here. It separates our personal experiences, our episodic memories, from just general context-free facts, which are semantic memories. I ate a sandwich for lunch yesterday is episodic. A sandwich is a type of food is semantic.
So a leading theory for how we store those episodic memories is called the factorization hypothesis. The idea is that the brain stores the what, where, and when components independently. They're thought to be orthogonal, which is a mathematical way of saying they're on separate perpendicular axes, kind of like length, width, and height. This would be a really efficient way for the brain to disentangle the content of an event from its specific timing. And we've got some pretty compelling evidence for this idea, coming from something called time cells. Just look at this heat map. You can see that different populations of neurons fire in these really reliable sequential patterns over a set period. Altogether, they create a kind of neural trajectory that maps out the passage of time for a specific event. This graph here really drives the point home. It shows just how accurately researchers can look at this neuronal activity and decode the when of an event. And it seems to be independent of the what that's actually happening. It's pretty amazing. But as elegant as this factorization model is, when we look a little bit closer at the data, it turns out the full story isn't quite so neat and tidy. There are a couple of significant challenges. Okay, if we go back and look closer at that time cell heat map, you'll notice something really interesting. The precision of the time coding isn't uniform. See how the signals get wider and fuzzier later in the sequence? They get less precise over time. And what's fascinating is that this change is logarithmic, exactly what the Weber-Fechner law would predict for our subjective experience of time. The first major problem is an issue of ambiguity. Is a particular neuron firing because a certain amount of time has passed, or because the subject is in a certain place? Well, it turns out that the answer can actually depend entirely on how the experimenter chooses to analyze the data. The same cell could be labeled a time cell or a place cell. And the second problem, well, this one challenges the core idea of the model itself. It's a fundamental logical error. The idea that space and time can be completely independent or orthogonal is flawed. You can't get from one place to another without moving, and any movement inherently takes time. The two are fundamentally linked. Okay, so if the model has these cracks, how can we fix it? Well, one potential solution proposes a clear division of labor within the brain. This model uses a great analogy: the librarian and the library. The idea is that the hippocampus acts like a librarian. It doesn't store the information itself, but it knows the order of things. It encodes the temporal sequence. The neocortex, on the other hand, is the library, where all the content, the what, is actually stored. Now, there is an alternative solution. It's a bit more complex, but it suggests that maybe these dimensions aren't separate at all. Maybe they're intrinsically woven together right from the start. In this view, a single population of neurons encodes all aspects of an experience at once. The what, where, and when are all mixed together. Their combined activity creates this complex, high-dimensional structure. You can imagine it like a twisted and folded sheet of paper. Scientists call this a manifold. So even though all the dimensions are tangled up in this single structure, you can still, in theory, separate them out later using mathematics.
And this all leads to a really, really crucial point. Just because we, as scientists, can observe a pattern of brain activity that correlates with a stopwatch, that doesn't necessarily prove that the animal's brain is actually using that specific pattern to calculate or to feel the passage of time. Correlation isn't causation.
All right, so let's wrap up part 1 with the big takeaways. First, our sense of time is a complex construction, not a direct perception. Second, the elegant factorization model provides a great starting point, but it's seriously challenged by that deep, unavoidable link between space and time. And it's this very challenge that opens the door to what we'll be talking about next, the idea that brain-body rhythms might just be the missing piece to this entire puzzle.